So now I'm going to discuss how we diagnose viruses in the laboratory. And so there are some classic diagnostic mechanisms, and then of course there's some more modern diagnostic mechanisms. So in general, because viruses infect cells, one way you can really look at whether or not there are viruses present is whether or not you have what's called cytopathic effect, or CPE. And CPE is just in essence a structural change to the host cells. So you can see over on the before, you have an intact, what we call monolayer. It's just a carpet of cells. They're all touching each other. They're happy. They're healthy looking. And then after infection, you can see that the nice carpet of monolayer is gone. And some of the cells are balled up. They're not touching anymore. There's not a confluent layer. And that's because your virus is killing the cells. They are lysing the cells, and the cells are dying. And that's referred to as cytopathic effect. Now, there is also hemagglutinin assays. And so there are some viruses that have hemagglutinin proteins, and these can cause agglutination of your red blood cells. So mumps, parainfluenza virus, and influenza viruses all have the ability to absorb heme. They have the hemagglutinin proteins on their surface. So we use antibodies to identify many viruses. And antibodies are going to be used in different types of reactions. So you have here, first we're going to talk first about our hemagglutinin inhibition assay. And so the inhibition part is going to be your antibodies. So as I mentioned before, you can do agglutination. There are some viruses that have H proteins on their surface, hemagglutinin proteins on their surface. And what basically happens is you add the virus, like an influenza virus, parainfluenza virus, and you add red blood cells, and the virus will bind to the red blood cells and cause clumping, agglutination. And you can actually visually see this in a test tube. It's pretty easy to detect. Now, if you add the virus and then a person's serum antibodies, and you let the antibodies interact with the virus first, and then you add your red blood cells, so this would be 1, and then you add your red blood cells 2, and then this will be the readout 3. You don't see agglutination because the antibodies are preventing the virus from interacting with the red blood cells. And so that's the hemagglutination inhibition test. And again, the viruses that this will help to detect are things like influenza and parainfluenza virus. Now, I just pointed this out that influenza is an orthomyxovirus, and parainfluenza, even though it has that name influenza in there, it's a very different virus. This is a paramyxovirus. This is the same virus family as measles, rubella, mumps. Parainfluenza virus is going to be able to cause upper and lower respiratory infections. It's the main cause of croup, which is going to be inflammation of your voice box, the larynx, as well as the trachea. So human parainfluenza virus 1 and 2 are the most common causes of croup. Now, another thing that you can use antibody for is like a complement fixation test. And so as we know, antibodies are in the serum, and they're able to, if they're IgG, they have, and IgM, they have complement binding sites. So they can fix complement. Complement will stick to them. And so you can use this to either detect an antigen or an antibody. It depends on the approach. So in this case, you have a serum with your antibodies, and then you add an antigen, shown here in these little diamonds, and it's going to bind to your antibody. Now, once the antigen binds, your complement is going to bind the complex. And then you're going to get hemolysin sensitivity with your red blood cells. And then your blood cells clump, similar to like that agglutination. And then you have no lysis of your red blood cells. And then in the case of your without antibodies, you have antigen that's free to float around, complement's free to float around, and then your red blood cells are going to get lysed because the antigen will bind to the red blood cells, which will recruit your complement, and then you'll get your lysis of your red blood cells. Now, a neutralization test is very straightforward. I don't have to say too much about this. Basically, you have a virus, and in this case, it's a pseudovirus, which means it's not SARS-CoV-2, but they just added the S protein, the spike protein, to the surface of the pseudovirus. And now this virus can infect ACE2 expressing cells. And then if you add someone's antibodies, you have anti-S protein antibodies, you are now blocking the interaction and the virus cannot enter the cell. So you're neutralizing the virus's ability. Now what happens to this virus? Well, remember, your antibodies have Fc regions, your macrophages have Fc receptors, so this virus will get eaten by a macrophage and will be eliminated. And then a fluorescent antibody. So you can use antibodies to light up a pathogen. And so in this case, we have a bacterium, and the bacterium on its surface has proteins, which our immune cells see or our body sees as antigens, foreign antigens. So we make an antibody against that bacteria protein, and these antibodies will coat the bacterium. And you can fluorescently label that antibody so that it glows. And so this is Legionella, and you can see that the antibodies are basically coating the bacterium, and then you can visualize this using a fluorescent microscope. Now another is a radioassay, a radioimmune assay. And so what's happening here is you're going to have a radioactive antigen. So basically you can add influenza hemagglutinin antigen at a fixed concentration, like 10 particles. They're only showing 8 here, so 8 particles. And so you have your antibody that is specific for your radioactive antigen, and this is called the first antibody. And then you're going to take the patient's blood, so a person that you suspect is infected with influenza. And you're going to take their blood and you're going to add their blood, which will have some sort of antigen that is specific for whatever pathogen is your patient's infected with. And so that's the unlabeled antigen. So remember, you added flu because you think flu is the culprit. So you add flu and it binds to the antibody, the flu-specific antibody. And that is labeled so you can detect it. And then you add the patient's blood, which you suspect has flu antigen. So it should be able to bind to that antibody just as well, but it's not going to be labeled. And then so what happens is your radioactive antigen gets displaced by the flu antigen that came from the patient, those blue balls. And then your unlabeled antigen, these blue balls, are going to start to bind better to the antibody. They don't have that radioisotope interfering with them. Then you're going to precipitate, and then you're going to look at radioactivity of the supernatant. So you can do this because what you're looking for is how much of my original radioactive antigen was displaced, and that can tell you whether or not you have a positive that the person has actual flu. If they were negative and had RSV, RSV would not displace the initial radio-labeled antigen. You can also measure the precipitate of the bound antigen, so you'd have to use a second antibody that's specific for that first antibody. And so that's similar to the ELISAs that we were talking about previously. And so next up is an ELISA. So basically this is a very straightforward test. You coat with an antigen, you add your primary antibodies here. So here's your primary antibody. Oh, here they are, I'm sorry. Here's your primary antibody. And then you add your secondary antibodies. Here's your secondary antibodies. And the secondary antibodies have the color, so you get a color produced. So again, you could be thinking that your patient in the past has been exposed to Ebola. So you take serum from the patient, you add it to a plate with Ebola proteins bound to it, The serum, if the patient has any anti-Ebola antibodies, will bind to the Ebola antigen. And then you'll have a second antibody bind to the human antibody that will fluoresce, and you can see whether or not your patient has Ebola antigen or antibodies. Now, what is seroconversion? Seroconversion is where you can measure an increase in antibodies because a person has been exposed to the actual pathogen. And the cutoff is a 4-fold increase. In your antibody titer. And this, of course, has to have a reference. And usually this is a normal population. An example is HIV. If you are seropositive for HIV, meaning you have HIV antibodies, equals you're HIV infected. So this is our example. So seroconversion means that you've seen the pathogen. If it's a known chronic infection like HIV, seroconversion means you have the infection. So it can be used interchangeably. Now, of course, we have a lot of PCR mechanisms, and nucleic acid mechanisms are very important because they're very rapid and you can try to detect many different pathogens at the same time. And so this is just an example of a PCR test. But basically, your PCR is going to have some sort of sample. So depending on what type of infection, it could be from a skin infection, it can be from urine, it can be from a stool, it can be from your nose, that's shown here. And then you're going to have to collect the infections, extract your RNA or nucleic acid of interest, and then you're going to do what's called reverse transcriptase RT-PCR, and you're going to use specific primers for this. So the primers and probes that you're going to use are going to be very specific in this case for COVID. You can have flu-specific primers, Ebola-specific primers, Epstein-Barr virus-specific primers. All the viruses will have different sequences, and the primers are going to be specific for those differences so that you can tell the difference between the different pathogens. And then you get a test result. So your test result will have some sort of threshold. Again, this is typically going to be a population kind of background signal. And then you're going to have your positive cases have to reach some statistical measurement above that threshold, and negative, again, has to reach a statistical non-detection below that threshold. So with COVID tests, again, there's going to be nucleic acid tests, there's going to be antigen tests where you're actually trying to detect whether the virus is actually there and replicating, so you're looking for virus proteins. And a serologic test. Did you have an infection in the past? Do you have antibodies? Have you seroconverted? Now, a seroconversion for SARS-CoV-2 is very different than HIV. If you have IgG, so this is a yes, for SARS, it means you were infected. There are very few, very, very, very few tests for IgM. Measuring IgM levels is tricky because they're bulky and you can get a lot of false positives with them. So typically, they are going to wait until you could measure IgG responses because those are much easier to measure. And then IgG comes up too late following a SARS-CoV-2 infection. So typically you've already cleared the virus by the time you have that IgG response. And so those are typically the types of COVID-19 tests you will have.